some of our research in a kind of a bird eye view perspective, not getting too much into details, but in kind of trying to convey what sort of questions we are trying to address in our basic science, basic immunology laboratory, which is located at Rockefeller University. So I like always to start these presentations with kind of a comparison in terms of textbook, what we learn in textbook immunology, and the way we like to see the immune system in our labs. And that is shared between several of the labs that are part of the FASI initiative. So this is on the left side. What you see here is basically what we learn in immunology textbooks, which is the immune system of our body is providing a resistance, which is combating the pathogens that are trying to invade our body. That's the main function of the immune system, as we learn in medical, in biology textbooks, as well as the way we see this in kind of in the press. My lab, as well as several other labs that are part of FASI, are studying the other side of that. We are studying how we are able to deal with another source of non-foreign antigens, of things that do not normally belong to our body, which are the diet and the microbiota components. So in contrast to pathogens, microbiota as well as dietary proteins are providing a series of host benefits that are important to maintain our body functioning, in fact. So instead of providing resistance or combating those antigens, those interactions, we need to develop another form of immune system function, which is called tolerance. That's the way that we can assimilate the interactions with non-self proteins that are present in the diet or the microbiota. Additionally, immune response triggered by pathogens may damage our own tissues. To deal with inflammation-induced tissue damage, we must develop another form of tolerance that's called disease tolerance. That's not going to be the focus of the presentation today, but that's a major focus of study also in the labs that are present in FASI. But overall, the overarching goal of my laboratory is to understand how the intestinal immune system balance resistance to the existing pathogens that are always in contact with our body, and the most common non-pathogenic interaction that we have with diet as well as microbiota. So these are the balances that we are trying to understand in the lab as a whole. Here you can see kind of an image of what we represent of the intestinal immune system as well as the intestinal tissue in the body. For those that are not familiar with these kind of textbook images of the intestine, here you see the intestinal tissue, which is formed by the single layer epithelial cells. That means that's the single layer of cells separating the core of our body to the outside environment. In this case, the outside environment of the intestine is the lumen of the intestine. That's where the food passes through. That's where most of the bacteria are residing. In the proximal part of the intestine, you have most of the absorption of the food and dietary proteins. As you go distal from the proximal part to distal part and towards the rectum, then you have accumulation and increase of density of gut microbes. So you have on the proximal part of the gut, proximal to the duodenum or the mouth, you have more food absorption. In the distal part of the intestine, you have accumulation of microbiota, of bacteria that are part of the commensal bacteria. The lab studies different aspects of this surface of the intestine. We study how the epithelial cells, as well as the immune cells that are residing there, can deal with this stimulation by non-self proteins from the diet, as well as microbiota. We study, as Chris mentioned in her introduction, the communication between the gut and the brain. That is not the topic of the presentation today, but that's a major interest of the lab. But we also study the initiation of the immune responses against luminal stimulation, against luminal antigens, against proteins from the diet, as well as microbiota antigens. And that normally takes place in these gut draining lymph nodes, which are a lot of them are present in both humans as well as in animal models that we study in the laboratory. But I like to focus today on the topic of the presentation is how do we avoid food allergies? But before getting to this topic, I like to remind the way that Ruslan Medzitov, which co-founded FASE with Chris, likes to explain the difference between allergic disease versus allergic defense. So allergy, as we learn in textbook as well as in the media, is thought to be a mistargeted pathological response to innocuous environmental substances. For example, food proteins, the way we react to peanuts or react to a variety of proteins that can cause food allergies. However, it's important to remind, for those that are not familiar, that allergic reactions exhibit all the features in terms of immunological features of a defense system that protects our body also from noxious substances. So all allergy symptoms, including runny nose, tears, sneezing, itching, vomiting, all are also associated when we ingest or get in touch with some harmful substance, for example, venoms, poisons, or rotten food, for example. So these are all common as a part of our body to defend and react and expel substances that should not be inside the body. And I think this kind of distinction between a normal allergic defense, which is in place to kind of eliminate noxious substance from our body, to allergic disease, which is what we see in food allergies. This is important to distinguish. So I'm just gonna kind of summarize in a graph what Ruslan means by that. So if you look on the x-axis of the graph, on the flat part of the axis, you have the response to substances. The more response you have, the more you go to the right side of the graph. And on the vertical part, on the y-axis, you have whether the effect is harmful to the body or beneficial to the body. So if you have no allergic response, basically, you may be cause harm. You're also going to have harm in the body because you may not be able to defend against this noxious substance that I was referring in the previous slide. In this square here, in this rectangle, that's when you have some response, but not the exaggerated response against the substances. That's when you have the benefit to the body. You have allergic defense, preventing noxious substances to get inside our body. But if you overreact against these noxious substances, for example, what causes food allergy, then you have also pathology. So there's distinction between what is a normal allergic defense that probably was evolutionarily selected to deal with the substances versus an overreaction of a normal body reaction that is causing allergic disease. I'm going to get now into another aspect of this discussion, which is the topic, one of the central topics of my laboratory. And that's the phenomenon of oral tolerance. I'm going to remove this floating panel here. So oral tolerance is defined in terms of immunologically as an active process of local and systemic unresponsiveness to orally ingested antigens. That means if we ingest a protein prior to another contact to this protein in an immunological active form, you prevent the development of this allergic reaction. So that's how, presumably, our body prevents seeing foreign proteins as something that we should actually develop inflammation. This phenomenon of oral tolerance was actually first described almost 200 years ago by Dakin, and this was very interesting because this researcher found that a common practice among American Indians was to chew leaves of poison ivy to prevent the painful skin rashes that we have when we get in contact with this plant. The important point here is that the chewing of the leaves should happen before you develop the skin rashes. So that's a form of preventing the kind of pathological reaction to this poison ivy plants. That is in fact, is still a common practice among several Indian tribes in America, Central America, North America. Subsequent studies, as you can see here in the timeline here, by Berezka, Wells, Chase, and so on, kind of try to bring to the research laboratory setting the study of oral tolerance. And basically, there are several examples. For example, oral or rectal administration of milk prevent anaphylaxis to milk injections. Feeding a vegetable protein prevents anaphylaxis reactions to this to this protein. And then in more recent settings, which I'm going to describe in the next slide here, in the 1970s, these researchers here that are listed here began kind of doing more systematic studies to understand how oral tolerance is initiated, how this is maintained, what's the relevance of oral tolerance in preventing kind of food allergies. And the way this was done is in the following, and I'm gonna not focus too much on this type of presentation today, but basically you have normally, if you have an exposure of a protein plus what immunologists call an adjuvant, that's basically what you put together with a protein to trigger a vaccine-like reaction to a protein. So if you give a protein, in this case of albumin, which is the protein of the white egg, of the egg white protein, together with an adjuvant, that induces a vaccine type of response, which is a strong response to this protein. That's how we normally develop the vaccination strategies. However, if these animals had fed this protein prior to this contact, then they prevent this inflammatory reaction to this protein. And that is basically the phenomenon of oral tolerance. It's important to point here that this is is exactly the opposite of what normally people are trying today with oral immunotherapy, that you give the protein orally after you develop an allergic response. I'm going to discuss this later with you guys in the Q&A probably. So as an example of how we do this in the lab, we provide animals with this protein orally or compared to the water lack of the protein. Then we expose these animals with an adjuvant, in this case, the cholera toxin, in the presence of this protein. If these animals had ingested the protein prior to the exposure to the cholera toxin, the adjuvant, you prevent the drop in temperature, which is an evidence of anaphylaxis that you see in the control animals that drank water. So the ingestion of this protein of albumin prior to the adjuvant or the immunization, vaccination to this protein prevents this deleterious reaction to this protein that leads to the drop in body temperature in the development of anaphylaxis. So that's a very powerful phenomenon. It's a very reproducible. It can be seen in any settings of the laboratory. So of course, what immunologists as a whole, many of us at FASE are trying to understand is what are the mechanisms that are inducing this phenomenon of oral tolerance. In other words, what is so special about the intestine is a tissue that is able to induce that? So if we go to the gut draining lymph nodes that I described in the first slide, which is a major topic of laboratory, that's where the initiation of the immune response to these luminal antigens, including dietary proteins, are initiated. That these are the sites that initiate an immune reaction against a particular protein. So long time ago, our laboratory, as well as others in the field, described that in the gut draining lymph nodes of the intestine, there are presence and induction of this type of anti-inflammatory immune cell called regulatory T cell, which is particularly rich in the gut draining lymph nodes. So the intestine seems to be very prone, very much able to induce these anti-inflammatory cells in these gut draining lymph nodes, which initiate the mechanisms of oral tolerance. How are these Tregs induced? That's the next question that people try to address. But first, if we go back to the scheme I showed in the first in the slide, a few slides ago, the feeding of the protein induced these anti-inflammatory cells that prevent, when you have the adjuvant, the immunization, the development of these inflammatory cells that would trigger food allergy. So the induction of the anti-inflammatory cell type prevents the development of the inflammatory cell type later on. How are these regulatory T cells induced? Again, we and others found that there's a particular cell that is called antigen-presenting cell, particularly the cell called the dendritic cell. That is the cell that mediates the development of this inflammatory versus regulatory T cells in these gut draining lymph nodes. And we found that the intestine contains a specific subset of these dendritic cells that are tolerogenic. That is, they are able to induce these anti-inflammatory T cell subsets. For those that are not familiar with T cells are basically cells of the immune system that are able to induce all the inflammatory or anti-inflammatory reactions against proteins. They also regulate, for example, the production of antibodies by B cells and so on. So these are central cells in the immune system. And in the intestine, what is interesting that there is a subset of these dendritic cells that are modulating the development of regulatory T cells. We found that these cells are coming from the intestine and migrating to the gut-draining lymph nodes to mediate the development of these specific regulatory T cells. And we found that different areas of the intestine have different propensity to induce tolerogenic cells, the anti-inflammatory cells, versus the pro-inflammatory T cells that are taking place in the distal part of the intestine. So depending on where in the intestine you analyze the initiation of the immune responses, you have different propensity to induce an anti-inflammatory response versus an inflammatory response. What is important in the context of tolerance is that, as I mentioned early on, the absorption of food proteins takes place in the proximal part of the intestine, and that's where you have more propensity of inducing this anti-inflammatory immune cell subset, the regulatory T cells. So what we are trying to understand in a laboratory is what goes wrong when you have an exaggerated response to food proteins in the context of food allergies. So we are trying to define what types of dendritic cells and T cells are mediating this response to food proteins. And the development of new tools that we have in the lab now have allowed us to identify subsets of cells that are mediating this exaggerated response to food proteins that leads to food allergy. So to summarize part of this topics of the laboratory here, if you look at the development of hypersensitivity, which is the development of allergies, is exactly the opposite of what happens during tolerance. During tolerance, you have a regulated immune response. You don't have an exaggerated immune response that allows concomitant absorption of nutrient absorption, digestive process, and maintains an intact barrier of the intestine. When this is regulated, and that's what we are trying to find the mechanisms, you have exacerbated immune response. You have primed immune cells that release, for example, histamine. That's the case of mast cells in response to antibodies like IG, and that leads to disruption of the epithelial barrier of the gut, and then the development of food allergies. So basically, to end the presentation here, we think that this understanding how the tissue in the intestine interacts with the neighboring cells within the gut to modulate the development of resistance against pathogens, as well as the tolerogenic response against food proteins. We think that we are able to understand how this process can be dysregulated during pathologies. So I'm going to end here to leave some time for discussion. And I think I start with Chris. So I'm going to stop sharing the presentation. Thanks for your attention.
Dan, this was such a great talk, and it's amazing the research that your lab has been able to do over the years. Very impressive. And then just the topic of tolerance. It's such the holy grail for food allergies in general, as well as probably a lot of other autoimmune diseases when we think about it that we believe arise from the gut. So very nice. So please put your questions in the Q&A. And I have one question for you here. What's the connection between, infections and allergies? When you think about when you had explained there's an adjuvant, right? Meaning something else that might trigger the reaction against the dietary protein.
Yeah, so that's a very important question because sometimes in the laboratory setting, we observe that depending on the phase of the infection, the exposure to the food protein can trigger more propensity to allergies. That is different, for example, when people probably are familiar with the use of helminth, of worms, to kind of suppress allergic response, for example, is used in asthma. That's because after the establishment, the infection with helminths may trigger anti-inflammatory cells that are able to dampen inflammatory reactions that is seen in allergies. But that is different than when you have, in the peak of the infection, the initial exposure to the protein. So in a given window, it's possible that a particular infection can actually trigger or boost an inflammatory reaction to the protein. So my main point here is that the timing is very crucial, as well as the type of pathogens. For example, helminths are known to suppress allergies later on. However, that is not observed against bacterial or viral antigens. For example, infections with bacterial virus are not known to suppress allergies, but also the timing when the infection is taking place in comparison to the absorption of these dietary antigens.
Yeah, no, that's interesting because there is some data out there suggesting both for asthma and food allergy, certain viral infections at the time with introduction of certain proteins can result in disease state. And I did like in the beginning how you had explained that allergic disease and allergic defenses is normal, right? And food allergies is an exaggeration of that doesn't shut off properly.
That's right, yes.
Very critical for understanding that these defense mechanisms are necessary. So why do people have an immune response, in their blood against certain foods or antigens, but then they never react? Like you see this a lot, you get the blood tests for food allergies. Someone says, I have a high IgE, but I eat that food. I'm not allergic to it. How do you think that occurs in this setting of tolerance?
Yeah, so this question of comparing similar patients with similar markers for allergy that develop a very different outcome, different response, it's almost the central question that many of us are trying to understand in the fasting. And we believe, as well as some of the people in the field, that this is related to kind of this gut-brain connection. It's possible that with the same amount of, for example, histamine or release of inflammatory mediators, different patients are able to deal with these inflammatory mediators in a different way, and that could be in part mediated by this gut-brain connection, because a lot of these reactions that we see in allergies are mediated by a neuroimmune interaction. For example, the vomiting, the rash, and so on. So if the way that the nervous system perceives this inflammatory reaction is different between patients or different conditions, that may lead to a kind of disparate outcome. And that's kind of the hypothesis to explain this, what you're seeing in the clinic, Chris.
Yeah, it's pretty amazing because the first thing you think about food allergies is IgE, but how complex it is with the nervous system involved in the gut-brain connection. We have a couple more questions here. What do we know so far about the requirements of the initial protein? You use the example of the egg whites treatment to induce that tolerogenic response, and how far in the gut does the protein need to get to induce tolerance?
Okay, so there's this first question. There's a lot of interesting points, and I'm going to try to be brief, otherwise there's no time to answer the other questions. But basically, the protein needs to reach the proximal part of the gut. We think that, in fact, we measured using radio labeling approaches. We measure how far the protein can reach. It can reach very distal, but the proximal part of the gut, the duodenum, is normally where you have this propensity of inducing tolerance. And it's important for this question, as well as for some of the other questions that come in later, the discussion of the immunotherapy, oral immunotherapy. And normally this is happening before, because you develop these anti-inflammatory cells before you develop the inflammatory cells. This anti-inflammatory can prevent the development of the inflammatory cells. That's why the timing is very crucial. And that's why the oral immunotherapy, you start with a very, very low dose and escalate over time, as opposed to the tolerance that can already come with a high dose. But I may have time to discuss this later on.
Yeah, just to add on to that, what do you think if you put in a compound, like a PPI, a proton pump inhibitor or something like that, that doesn't allow for the breakdown of those products quite as much, or assuming they don't, could that influence, the protein evaluation at all?
Yeah, so that's very interesting. In fact, we have tested this in the lab. In some settings, you may not want protein to be absorbed. For example, if you have particular infection that is happening in the place that the food should be absorbed. So we modulated that, and by modulating where the food is absorbed, by using different drugs, including protein pump inhibitors, indeed you affect the efficacy of the tolerance induction. So ideally, it should be happening in the proximal part of the gut. The more you go distally, the less efficient the tolerance is.
Mm-hmm. So, and here is very good scientists, Dr. Slattery, question. Evolution of TLR biology continues to be very conserved and gradual. However, antigen from food sources are rapidly evolving due to engineering. Can we reset this tolerization in the face of rapid re-engineering of antigen presenting?
Yeah, so William asked a very interesting question, and it's kind of underlying question that we are trying to understand when I mentioned the types of dendritic cells that are modulating the T cells. And in fact, there is a person, there are two people in fact that are studying kind of the change, the rapidly evolving source of food antigens. One of them is Andrew Wang, that is studying, for example, how the presentation of the food protein with xenophobes that are present in today's life can affect how efficient this tolerance can be induced. I'm not going to get too much into that, but basically to answer what William is saying, TLR biology, that's are receptors that are known to bind pathogens, and this is very conserved in evolution. And indeed, food sources are changing quite rapidly, and that may trigger, the appearance of the xenophobes that actually xenobiotics, not xenophobes, xenobiotics that are substances that are normally not present in food sources. And the xenobiotics are actually can be affecting the tolerization aspects of these proteins, and understanding basically what cells are kind of being activated by these xenobiotics in different conditions, which is part of the question. It may be able to allow us to target different pathways that are triggering these cells, so we can kind of restore tolerance that may be lost in some of these cases. I think that's kind of the underlying reasoning that William asked.
No, that's
both are very bad. Xenophobes as well as xenobiotics. So.
Yeah, so all these unnatural chemicals in our diet are not very good for us.
That's right.
And Andrew Wang did present in a webinar not too long ago that's on our website, showing that some of the common preservatives in stable shelf food can result in more sensitization as well. Someone asked a little bit of let me just see. Do dendritic cells that carry allergen travel to the distal lymph nodes that mediate inflammation?
Yeah, so that's a very interesting question. So basically the underlying question is, because the proximal part of the gut is where you develop more tolerance. In the case of allergies, is that because the dendritic cells are traveling to the distal part or to the distal drain lymph nodes? We don't know the answer for that yet. But what we are doing is to compare antigen presentation by dendritic cells in the presence of the so-called adjuvants or absence of adjuvants, and see if the difference is the state of the dendritic cells versus the location. And basically, we don't have an answer for that, but that's a very interesting question.
Yeah, and someone asked, can you speak about, and you had briefly mentioned this in one of the slides, about the oral immunotherapy desensitization, where you give a little bit of allergen to someone to kind of suppress the immune system, and thoughts on using that to build tolerance to allergens? Yeah,
so I think people that are familiar with allergies probably know the pros, of course, is because you are using kind of the food itself as a treatment. You're not using any drug that may have off-target effects. But the cons is also a part of that. First, we don't know exactly how oral immunotherapy works in terms of cellular mechanisms. It doesn't work for everyone. The later, the older you are, the less efficient oral immunotherapy works. And it doesn't work even in young age for all of the patients. And I would say that it's possible that by trying to make the parallel that I was trying to make here in terms of basic science, in terms of understanding how oral immunotherapy works, that we can make it more efficient and work for a larger range of patients, which is the current kind of somewhat limited. But the first drug approved in many decades, Chris, you mentioned this other day, is actually an extract of peanuts. So that's pretty. It shows that the field needs to advance.
Yeah, the one and only FDA-approved therapy for food allergies is palfozoria, which is a peanut pill. And so what we don't know is, how sustained that would be, the desensitization. There's a lot of different interplay with it. And if you stop the allergen for 2 years, but then you start reacting again because you have these memory cells, and there's never been any study looking at that. Well, we are out of time now. We're past our 12:30 mark, but thank you so much, Dan. It was a great talk, and your research always fascinates me, and I love hearing about it. So thank you so much. And FOSSI has single-handedly transformed how we diagnose, treat, and prevent food allergies, and we will not stop until we find a cure. Scientists and advocacy groups all over the world are watching our science, which is solely funded by generous donors. Donate now at foodallergyscience.org. 100% of your donation goes directly to the scientists' research. Thank you for making our Food Thought webinar series a success. We'll have an exciting new webinar lineup for you in the fall. Until then, enjoy your summer and keep on imagining a world without food allergies. And FOSSI will keep building it. Thank you. Thanks, Dan.
Thank you.
Good day.